Good morning, or good afternoon, depending where you're at. Welcome to day one of this forum on unplanned clinical trial disruptions. I'm Jim Rosenberger, Director of NIS, and I'll be the host for this event, along with Glenn Johnson, who will help with any technical issues. We now have over 100 members joining us, and we'll hopefully get a few more before we begin. Today we have three experts speaking from their experiences in government agencies, and next week we have three additional in the day two of this forum. If you have questions during the presentations, there's a Q&A box at the bottom. Please use that to submit your questions, and we will provide them to the speakers during after each session or at the end of the three presentations. I'm now pleased to introduce Nancy Flournoy. She's chair of the Ingram Olkin Forum Committee for NIS, and she's a founding member of the NIS Corporation, and now University of Missouri Curators Distinguished Professor Emerita. Nancy, I turn it over to you.
Thank you, Jim. I'm delighted to be able to help present this forum for NIS. So I am going to share my screen. So I hope everyone can see this. Welcome to the forum on unplanned disruptions in clinical trials. I want to say a little bit about the Ingram Olkin forums in case you haven't heard of those before, and then a bit about this series of sessions that we've planned for this particular forum. So the Ingram Olkin forums are established to honor the memory of Ingram Olkin. And we have the tagline for them, Statistics Serving Society. If you are too young to have heard of Ingram Olkin, or you've been living in a cave for the last few decades, you can Google him and find lots. And if you don't know him, you should. And if you do know him, you know we could talk for hours. And instead of doing that, we are going to jump right into this session, which is really honoring his devotion to the statistics as a profession and to statistics' role in society. So the forums are, the forums in general, of which this is one, are established to consider a current social issue, and not just any current social issue, but one where statisticians have not been largely engaged previously, and try to bring together people with the issues and statisticians to develop, to find those statistical problems that need solving, to develop statistical action items, to move the science surrounding the issue forward, and to facilitate new collaborations. And so that's the background for this and the other forums. Now, we had our first forum in 2019 on gun violence. We had a number of very interesting sessions, and there will be an invited panel on this topic at JSM next week on Monday at 1 o'clock. So if you are interested in that, please do attend. There's been a lot of spin-off activities from that forum. We had a session, we had a forum on opioids planned for last May when the COVID epidemic hit, and so it has been postponed until next year. And we have one on artificial intelligence, the ethics of that, and bias in algorithms, particularly in finance and social justice. And that is just preparing to transition from a in-person forum to a online forum. So this is our first online forum, and we set it up as a series. So it's it's like, this is the first session, and the second session will be on July 28th. And these two sessions involve the perspective on clinical trial disruptions from the government agencies. And the primary goal for these two sessions is to motivate to provide you with information and insights from the government, obviously, or the government spokespeople, our experts, but also to stimulate your interest in the technical issues that we'll talk about later. And the following sessions will occur after JSM. And the main topics we've identified are estimate issues, missing data, and several varieties of adaptive designs that might be addressed by group sequential methods or sample size re-estimation. And you'll hear more about that after these sessions. So I'm going to work from the back forward now. Next week, same time, same place, we'll have Dr. Lily Yu, who is from the FDA, David Williamson from the CDC, and Aldana Rosso from the European Medicines Agency. And so we hope when you registered for this forum, you've also registered for the next one, so you're signed up for both. You can save the link. After the two sessions from the government perspectives, we will send out a survey. And one thing is to get your feedback on these sessions, but the other is to gauge your interest in the sessions that are going to follow, the more technical sessions that are going to follow. And depending on a few things I'll explain, there may be some participant screening at that time for the following sessions. So again, the original idea of these forums was to have them rather small and rather intimate so that in fact we could encourage collaborations. And it's not clear how successful we'll be doing that in a online format. But we intend to have some speakers and then a much longer discussion period than we have today, where we will break out into discussion groups. And to make that successful, we need motivated and moderators of these groups and other participants, which I've listed here. So we will encourage you to request a role, and how many people we can accommodate will largely depend on how many step up to take a leading role in this activity. Again, we expect to end up with a report or a manuscript afterwards and some new collaborations on new projects. So back to today, on the behalf of the organizing committee, I'd like to invite you to enjoy this initial series, the initial forums on we're developing the language here, the initial session in the forum series. And we have on the panel Adam Lane and Sergey Tarima will be representing the organizing committee. You'll see them on the screen, and they'll be helping to organize the questions and answers. So I will now turn to today. We will start with Nancy Geller from the National Heart, Lung, and Blood Institute, and followed by Dean Fullman from the National Institute of Allergies and Infectious Diseases, and finally Lisa McShane from the National Cancer Institute, and then we hope to have a lively discussion. So with that, I will stop sharing my screen and turn the screen over to Nancy Geller.
Good morning, everyone. I am now going to attempt to share my screen. I have to go.
You got it, Nancy. Just make it full.
Yep. Go full screen.
I have slideshow, but it's
There was that
not.
little Control L
Little podium button,
or
right?
Command L if you're on a Mac.
Podium button.
Yeah, lower right.
Yeah, right there.
Thank you. Great. Okay. So I'm going to speak about examples from NHLBI-sponsored trials. And in contrast to what Nancy F. just said, the views expressed here are personal and may not reflect the views of NHLBI. I want to thank Nancy F. for inviting me. I don't know if I qualify as an expert, but I'm going to try. Okay, I want to begin with a word about Ingram Olkin. At the time I made these slides, there was not even a picture of him on the website. So Ingram was a professor of statistics at Stanford, and he's known for many statistical contributions, especially to meta-analysis. But it's worthwhile mentioning that he was a wonderful mentor, especially women. And one reason for this forum named after him is he was a very great mentor of Nancy Flournoy, but he was a mentor of many of us. And this picture was taken at the NIST reception at JSM in 2012. He just was a wonderful man, and I'm very pleased to be participating in this forum. So to the topic, I'm going to perhaps ask more questions than I will give answers. I'm going to begin with factors affecting trial disruption due to a pandemic. And my 4 questions are: when did the disruption occur? And how far along was accrual in a clinical trial at that time? How does the nature of the intervention affect the trial disruption? And how will we modify the data collection and analysis? I have 2 examples. The first is a behavioral trial where accrual is completed, but the intervention is continuing. And the second is an example of a bunch of treatment trials with long follow-up, which are from the Bone Marrow Transplant Clinical Trials Network. So when did the disruption occur? Well, really, it makes sense to establish a fixed date, and this will make the data analysis easier. We use March 10th to 2020, and this is when accrual in most of the NHLBI halted. Now, some trials have reinstituted accrual and visits by now, but whether or not these are actually happening is center dependent. So how far along was accrual at the time of the disruption? The easiest case is when accrual is complete and only follow-up is needed. It's harder and harder as more accrual is needed. And the reason for this is that it's difficult to restart accrual after a trial has been interrupted, no matter what the reason. So if accrual is, suppose, 80% complete, maybe that will suffice. One has to reassess the power and the increased difference to be detected and agree that that's sufficient. This will, of course, require a protocol amendment, but it's less trouble than restarting accrual. And note, if accrual has lagged all along, tolerance is probably not the best strategy for the funding agency. I find that DSMBs are very tolerant. They say, well, there's a pandemic, let's wait another 6 months. But in fact, it's not the best strategy for the funding agency if accrual has been lagging terribly all along. But the real problem is for those trials which are midway in accrual. Accrual will be restarted, but it may be slow or unsuccessful altogether. For funded trials in the planning stage on March 10th, these trials will start, but problems with accrual are perhaps more likely than in the past, and accrual has not been the easiest thing for our trials all along. And I guess I pessimistically have to say that clinical trials may never be the same again. So the nature of the intervention is something that the disruption affects. So the first question is, can virtual visits substitute for real visits? And if a trial requires multiple patient visits for assessment, even with virtual visits, delays and missing data, which is not at random, will be likely. You have to be flexible, for example, with visit windows. So we have a date for the 6-month visit and it's within plus or minus 2 weeks. We need to be flexible about that and allow a wider window. And in any case, these middle visits may not be made, and we have to focus on the primary endpoint to try to get an answer to the trial question. So this is the biggie. How will we modify the data collection and analysis? And these are the questions. Is completion of the trial actually feasible? Should the sample size be decreased? Will the primary endpoint have to change? Will that be sufficient for purposes of answering a trial question? What kind of sensitivity analyses will be done? So I'm not going to spend more time on this in general, but do it through 2 examples of disruptive NHLBI trials. The first one is in a prevention trial or type trial. It's called Plan with Families. It's a primary care pediatrics trial. Plan is primary care, pediatrics, learning, activity, and nutrition. And the second example is a set of trials from the Bone and Blood and Marrow Transplant Clinical Trials Network. So in a sense, Plan is a really good example. It's a diet, briefly, it's a diet and exercise intervention for overweight children and 1 overweight parent. And these pairs were randomized to counseling or usual care. Treatment is 26 or more counseling sessions over 24 months, with the second year being quarterly counseling rather than more frequent. Weight and height are recorded at baseline, 6, 12, 18, and 24 months. The primary endpoint is a change in BMI from baseline to 2 years in the child. So at the disruption, accrual was complete, but counseling was continuing. And what this trial has done is introduce virtual counseling via Zoom or WebEx. And it turns out that this was easier for the subjects because they didn't have to travel to a site. And in fact, some who had not shown up for appointments participated again after not participating for a while. The other thing this trial is doing is focusing on the primary endpoint. And to get the primary endpoint, subjects have been supplied with scales and height charts, and they're watched while they are measuring. So Plan also is collecting COVID-19 related data. It's using something called the COVID-19 Exposure and Family Impact Survey. This is from the Center of Pediatric Traumatic Stress, and the URL is at the bottom of the slide. It's designed to be used in ongoing and new studies where COVID-19 may influence study outcomes. Conceptualizes exposure to the potentially traumatic aspects of COVID-19 and assesses the impact of the pandemic on the family. It should be completed by caregivers. And it's very simple. It's 25 yes/no questions about COVID-19 exposure and 12 additional questions on a Likert scale on the COVID-19 impact on family life. And then there's an open opportunity for additional comments. It's scored in a very simple way, and Plan has decided to collect these data only once because COVID-19 is not the primary focus of this trial. So there are other questions that are under consideration, and this is a work in progress. And one of these is from the CDC COVID-19 Community Survey Question Bank. I put the URL on the bottom of this slide. It has such questions as, have you been sick in the past 30 days? Have you seen a doctor? How long after your symptoms? Were you tested for COVID-19? Were you tested for flu? Have you socialized in groups in the past 7 days? Starting today, how long would you be willing to avoid certain activities? For example, restaurants, socializing with people over 60, and so on. Questions on how has this affected your work? Have you lost your job? And things like that. And knowledge questions where you select all that apply. To the best of your knowledge, which of the following can protect someone from COVID-19? And one of the answers is drinking bottled water, which I don't think is very relevant. So there are questions that do protect you, such as hand washing for 20 seconds and so on. But as I was putting together these slides, I realized that there's really something very important that we haven't considered in Plan. And that is for treatment and outcome information, we're relying right now on adverse event reports, which are free-form and really may not give us the information we need. And specifically, if illness occurs, does it impinge on getting outcome data? This we have to consider, and we need some more detailed questions to answer how COVID-19 is affecting the primary outcome of this trial. Now, we're fortunate in that these are parents of young children, adolescent children. So fortunately, this is not a high-risk age group, but some of the participants, some centers are from low-income areas, and this has implications for risk. So we're not done, and maybe it's not so simple. The BMT-CTN is a more complex example. It's a network that conducts hematopoietic stem cell transplantation clinical trials. And many trials occur simultaneously. So on March 10th through 20, accrual was put on hold, except for one aplastic anemia trial, which actually was lagging in accrual and has now decided to end accrual at the end of August. So accrual and in-person visits resumed earlier this month, but it depends on the center because some centers don't feel prepared to resume in-person visits and whether the patient was willing to be seen. So the first thing we had to do was modify the forms for visits related to COVID-19 infection. So this is a screenshot form. So did the visit occur? If yes, record the date. If yes, select the visit type, clinic visit, telemedicine visit, phone contact, more than one. Was the visit delayed? And if yes, select the reasons. And under patient illness or injury, there are more questions specifically about COVID-19. So we had to make decisions about BMT-CTN data collection. And the first thing was to say we cannot deny patients to enroll in COVID-19 clinical trials. Although in general at the BMT-CTN, we don't allow patients to co-enroll in other. But here we felt like we had no alternative but to allow enrollment. And further, we had to modify the BMT-CTN forms to collect COVID-19 data. And this includes additional questions to capture infection prior to the start of the preparative regimen or infusion, additional questions to capture COVID-19 infection after transplant, treatment for COVID-19, status of the COVID-19 infection at virtual visits. And there's an option for COVID-19 as both a primary and contributing cause of death. And this is very interesting because it turns out that cause of death may well be in the eye of the beholder. And we have that in general. But in fact, in BMT-CTN trials, all-cause mortality is generally included in the primary endpoint. So in BMT-CTN trials, the primary endpoint is usually a composite. And the example I'm using is the first occurrence of chronic graft versus host disease, or relapse, or death. So it's a complicated composite. And what we've decided up to now is that the primary analysis will remain the same as originally planned, but there will be extensive sensitivity analyses. And of course, trials will take longer and accrual will be slowed. So here's some modifications of a data analysis plan and the sensitivity analyses. So the first suggestion is to censor patients at their last assessment for those who have missed an assessment attributed to COVID-19, or for those who have telemedicine or phone contact. Second idea is to censor patients at the onset of COVID-19 infection. Third is to censor patients at the earliest of those two. Fourth is to censor patients as of the start of the COVID-19 pandemic. But this is only feasible for a trial in which most patients are in the late stage of follow-up or who have already completed follow-up. And the fifth is adjust for the COVID-19 era pre and post as a time-dependent covariate in a Cox model, rather than censoring at the COVID-19 pandemic start, to account for potential changes in baseline hazard rates pre versus post COVID-19. Now, these censoring analyses assume that the censoring mechanism is independent of the primary outcome. We don't think that's really true. If there are a sizable number of such censoring events, and there's evidence that the censoring is not independent, we have to consider something more marginal structural models with inverse probability weighting strategies have been suggested. Analysis here is an open question. The goal of the sensitivity analyses is to assess robustness with the primary analysis. Now, nobody promises you that these are going to be consistent, but the impact of various censoring schemes on the number of events and the width of the confidence intervals will need to be considered. And it doesn't matter what you do, interpretation will be an issue, especially if results are not consistent across all the sensitivity analyses. So in summary, the expected interruption will affect ongoing clinical trials, less so for trials with accrual completed than for trials with accrual interrupted. Trial completion may well be a problem for trials in the mid-stage of accrual, and really nobody knows what will happen. In any case, data analysis will have to be modified, leading to new and challenging statistical methodology questions. Collaborators in making these slides, both from the BMT-CTN statistics group and the plan investigators. Thank you.
Thank you, Nancy. That was a wonderful start and overview of issues. And in the interest of time, we will save questions for the discussion period. And I will invite our next speaker, Dean Fullman, to share his screen.
Thanks, Nancy Florney. So you can see my screen?
Yes.
Okay, great. Thank you. Yeah, I didn't know Ingram Olkin, but I've read some of his work and I know he's a giant in meta-analysis. And so I'm happy to be part of this. And I would like to thank Nancy for inviting me to speak here. I'm going to talk about sort of a couple of examples and sort of a methodological way to deal with unplanned interruptions, always sort of taking as a reference, what do we really want out of a clinical trial? and what is the great advantage of a clinical trial. So, trial disruptions, these happen, even before the pandemic era. And with the disruption, somehow we think that the normal procedures of the trial are not possible, whether it's follow-up or getting drug or recruitment or accrual. And with these challenges in design, this can motivate adaptations or design changes. And particularly with the pandemic, we're considering, I think broadly speaking, more profound changes than would be typically done with trials in the pre-pandemic era where things run more smoothly. And there's a lot of different ways that the trials can be disrupted and a lot of different ways you can try and change them. And I think it's important to realize or recognize we should also respect the basic tenets of clinical trials, which to me is basically randomization. I think they're the greatest medical invention ever. And so this has led to greater knowledge and greater cures and improving public health than anything else. So I think of randomization and respecting it as sort of the North Star of what should govern us when we're contemplating changes or trying to respond to disruptions in clinical trials. And I just sort of list a few key elements. You want to keep the group pristine as much as you can, and so there's not bleeding of interventions in the two groups. You want to keep the interventions as clean as possible, and you want to keep the outcome pristine as well. And then just a couple more perspectives. This gives you an opportunity to be creative to the specifics of your trial that's being disrupted and also being responsive. And so really we're led into the arena of unplanned blinded adaptations or unplanned adaptations for which we should try and remain blinded. And I want to talk a little more about that.
Do you want to scroll your slides?
Yeah. Oh, you're not seeing them scroll. Do you see example number 1 now?
No, we're on trial disruptions.
Oh, great. Okay. Do you see the full screen now?
Yes.
I've advanced to respect randomization with the North Star in it.
Nope.
No.
We don't see it.
Try page
Okay.
down.
I just did that. Do you see example 1 now?
No, just it's the second slide, your second slide.
Maybe unshare and then share again.
Thank you. I'm getting my own disruption. And thank you for helping. How about this, malaria?
Nothing yet.
It says start screen sharing, double click to enter full screen mode. Or
So it says on my screen, it says I'm sharing.
Yes,
it says I'm starting.
It says you're starting.
First screen, trial interrupted.
No, it doesn't show, it's just black though. Glenn, you need some advice.
So
Plan B is we could share your slides and you could say,
yeah.
But give it one more try if you can.
I will. Maybe I'll share my.
Share your desktop.
Yeah, that's what I was thinking. Yeah. Yeah, I'm pulling up my PowerPoint.
that looks, we can see that. That looks good.
And do you see trial interrupted now?
Yep. And let's go to full screen again and try that.
Oh, that's scary, but
Yeah, or you could just go through your slides using the way you're
yeah,
doing.
do you see respect randomization now?
Yes,
Yep.
yes, we do.
Okay, that's good enough for me. Okay, so anyway, randomization is great. We got to follow it, honor it, and every change, adaptation we make should try and preserve the integrity of the randomized trial. So I'm going to give a couple of examples now. The first one is for malaria, monoclonal antibodies. So I'll give a little background about the disease. Malaria is one of the top 3 infectious diseases in the world. It's a parasitic disease spread by mosquitoes. So the mosquitoes bite you, they give you parasites, and the parasites go through your blood, causing fever and death, particularly with young children. And to counteract this, young children in malaria zones will receive drugs every day to prevent malaria disease. So every day they have to take it, and you know, it's kind of a pain, especially if you're a kid. So it's inconvenient, and there are issues with non-compliance with this daily prophylactic regimen. Monoclonal antibodies are a different way of achieving the same end. So your immune system, when it's confronted with a pathogen, say malaria, it'll make antibodies to block or abort it. And in the laboratory, we can make clones of these antibodies that are specific for malaria. So we can grow them up in vast quantities and then inject them into children, say at the start of the season. And if they are properly constructed, they'll last a long time. So one administration and seasonal-long protection. So that's the promise. The plan was to do a trial in Bethesda to see how much dose you need and how efficacious they were, and then do a field trial in Mali, in a village that NIAID has in Mali. So anyway, on the right-hand side, I've got the malaria life cycle, which is kind of beautiful. Here, we start up at 1 where the mosquito infects these sporozoites, the parasites. I got some blue antibodies here to indicate that these are around, it's not to scale, but they would latch onto the sporozoites and prevent them from going into the liver and thus aborting the infectious process. So, you know, when you're doing these studies, you don't know how much dose to get. And so we were going to start it in a graduated way, starting with 5 milligrams per kilogram, 20, and then 40. And one thing we do in malaria research is what's called human challenge studies, where we actually give the volunteer malaria parasites and see if they develop parasites in the blood or not. This is sort of this has been a well-established model, and the main reason is that we have drugs that'll completely obliterate the parasites in the blood. So you can sort of see whether in fact antibodies have worked, and if they haven't, you can sort of cure the patient with drugs easily. So anyway, this is a schematic of the trial. We would give them antibodies and at some point challenge them. The different doses would have different amounts of circulating antibodies at the time of challenge. And then we can do a logistic regression. And this is sort of made up data, and it's a free form curve, just meant to show you sort of at a high level what we would do. So we might fit a curve to this and then say, okay, at this target level here, very few of the individuals get infected. And so this is a level of antibody we want to have and maintain throughout the season for the Malian children. So it's an important study to do to get this target, basically. So things were going along great. And then we had several of the dose groups, all of the dose groups enrolled actually. And then COVID-19 hit, so we couldn't do the challenge at all. So what to do? So this is an opportunity to be creative. And so what we did was for the 2 lower dose groups, we figured there would be like hardly any circulating antibodies when we restart the trial, which is going to be in the fall. And so we're going to redo the trial in the fall with the same 3 dose groups, 40, 20, and 5, but we'll also include another group that was originally given 40 milligrams, but a long time ago. And so that'll give us more variation in the amount of antibodies at the time of challenge, give us more information. And so just looking at that curve here, I've sort of schematically shown the February cohort, when they get challenged around in October or so, probably gonna have low antibody levels, high infection rate, but it can still help us estimate the overall curve. So that's good. So that's one example, sort of a major design change in a way, but it's sort of an obvious thing to do, I would say, or an obvious opportunity that was used. The next example I want to give is about HIV prevention. So this is a large, one of the issues, central issues in HIV prevention now is how to make it more reliable and have more of a population level impact. So we have drugs, there are drugs that are used to treat HIV disease that you can give before people are, when people are uninfected but at high risk, and these drugs basically prevent infection. They're quite effective if you take them. And so now we're into the second generation of this enterprise of HIV chemoprevention, and there's a large randomized study about 3,000 women who are at high risk of HIV infection who are going to get an injectable drug called cabotegravir every 8 weeks, or they'll get a daily drug, kind of like the malaria situation in some level. So this study was planned to have accrual for 2 years and have a minimum follow-up of 1.6 years, so that total follow-up would be 1.6 to 3.6 years. We were going to use a Cox model with a treatment indicator and to be stratified by site and do a standard intent to treat analysis. So that was the plan. And here's sort of a schematic of women enrolling at various times, and then some of them getting acquiring HIV or getting HIV infection. And here's a schematic of when COVID-19 hits, and you know that red thing goes off into the future, so I don't know when that's going to end. But for our trial, this would be the period where COVID-19 is an issue. Now that's sort of at the macro level, basically. The sites themselves had periods where it was hard to get drug, and so the women in the study were sort of deprived of the intervention. So that leads to a logical kind of analysis, which would be to do an adaptation based on when drug was not available. So the plan is to define these, what I'll call blackout periods, where the study product was not available, and define these periods so then they could be used later in an analysis. And importantly, you don't want to sort of pick blackout periods where one arm is doing better than the other. That would be, you know, a terrible thing. It wouldn't be respecting randomization. And so to ensure against that, so there's no question, you would do this, the plan is to do this with a blinded adjudication committee who'll say, yeah, there was no drug here from April to June, so that's going to be the blackout period for both arms for that site. And so then you can analyze the data. A real simple version of this, we're going to use, the plan was to use a Cox model, but a simpler version of this, just to convey the concept, would be 1 minus the number of events on, say, one arm divided by the total follow-up on one arm. And so during the blackout periods, I'm not counting events and I'm not counting follow-up, but in an even-handed equal way for both the injectable and for the daily drug. And this would be the estimated treatment effect. In fact, what we're doing is, as Nancy mentioned, one of her, I think, 4 or so sensitivity analyses was to, I think she talked about this, to remove volunteers or have blackout periods, basically. And there's an easy way to do this with the Anderson-Gill formulation, the Cox model. The last thing I want to talk about is FDA guidance on adaptation, or what we can do. So FDA has these great guidances, and they have one on adaptive designs, and they say in general, pre-specified adaptations should be okay, but COVID-19 wasn't really pre-specified. Fortunately, there's this really great paper written by Martin Pausch and Michael Proshan up on the left there that talk about unplanned adaptations before breaking the blind. And this is a paper you should read. One of the key take-home points for it is that if you're really serious and you want to be, rigorous about controlling the type runner weight, a permutation approach for inference will be a way to ensure you have rigorous control. So I want to run through a quick example of this. So this is not for COVID-19, but it sort of was related to a real world problem, which I think shows the beauty of this permutation correction, basically. So here's a crossover trial. A guy's randomized to get treatment followed by control or vice versa. And I'm going to call the difference the random variable, the period 1 outcome minus the period 2 outcome. So my data is going to be for each person, the difference in period 1 and period 2, along with the indicator of which treatment, of the order of the treatment, basically. So here's the data, and here's the treatment indicators or the order indicators. And what you can do is a one sample t-test using the treatment minus control difference. So I write it in this kind of complicated way when it's really just treatment minus control. But I want to write it this way with the sequence indicator because I want to, blind that later and show what can happen. So here's a blinded adaptation. It should be okay with the FDA or generally okay. So let's suppose I take my crossover data and split it up into 2 cohorts, the first and the second cohort. And I don't have those Z's, I don't have the treatment label, so it's blinded. And then I can calculate the sample variance for the first cohort and the second cohort. And I might think, gee, the smaller variance means I got a more efficient procedure. Why don't I do that? So I'll come up with a blinded adaptive t-test, which I'll call a BAT. And so I'm going to take the overall treatment minus control mean as my numerator, and then in the bottom, use the smaller variance and compare it to a t-distribution. Now, this is kind of cheating, and it's not what you would do. But you can show that there's an advantage with doing this procedure. And here's an example. If I have a sample size of 20, I have 2 cohorts, I can get a type 1 error rate of 0.08. More cohorts, more type 1 error rate, in fact, inflation. And you might think, Dean, yeah, but this is like so contrived. But this was really motivated by a real world setting where a person was evaluating a randomized trial, and there were a lot of different statistical methods you could use, all estimating the same quantity, but some were more efficient than the other. And the way you measure efficiency is looking at the variance of the estimator for the different methods, and then you pick the one that's most efficient. So it makes perfect sense, but in fact, it's really sort of an opaque version of this sort of blinded adaptation where you're picking the smallest sample size. Anyway, if you do a permutation test, the fact that you've picked the smallest sample size doesn't really matter. All it does is to scale the mean difference test statistic, and so you get no advantage with the p-value. So here's a specific example where permutation gives you correct inference, even though you did a blinded adaptation. There's another more subtle example here, once again, where permutation comes to the rescue. So let's suppose I have a study where I have an outcome, I measure drug level, and I have the treatment indicator, and I'm going to compare the outcome in the 2 groups. But I want to do a blinded adaptation, so I cover up the treatment indicator, and then I use drug level as a proxy for that. So I think I'm so clever and so sneaky because I'm sort of, getting away with something. And so you could do different blinded adaptations. You could say, I'll do 17 tests using this blinded approach where I'm using drug level in lieu of the treatment indicator. Or I could say I'm going to double the sample size if the t-test using the drug level had a p-value greater than 0.05. Both of these can, inflate the type 1 error rate. The joke is sort of on the person trying to be sneaky though, because the permutation test you're actually doing is that the treatment has no effect on outcome and drug level. So you're sort of left with an unappealing conclusion that, yeah, there's an effect, it could be an outcome, it could be on drug level, I don't know which. So the reason I like this is because, in that one example where someone was thinking, yeah, I want to do an efficient test, so I'm going to think of different things to do and pick the one that has a smaller variance. That was some clever thinking, and yet it could lead to type 1 error rate inflation unless you did a permutation procedure, which would sort of automatically control for it. And so anyway, I'll finish. Blinded unplanned adaptations. So this pandemic is going to lead to, I think, many unplanned adaptations. I gave a few examples for malaria and for HIV prevention. The key tenet is our North Star should be that we want to respect randomization and be creative. And then if you're doing this, you should consider using permutation tests for inference, and you should definitely read this paper. It's very accessible and very sound advice, easy to read. So with that, I'll stop and pass on my screen to the next person.
Thank you, Dean. And next is Lisa McShane from the National Cancer Institute, who is sharing her screen now. Lisa.
Lisa, you're on mute. Lisa? I see you're muted.
Lisa, we can't hear
Okay,
you.
there we go. Hopefully you can hear me now and see me.
Yes.
Great.
Okay.
Welcome.
All right. So thank you to the organizers. I think this is going to be a very stimulating discussion. I'm learning just listening to the other speakers. And I think we all have our unique challenges depending on the kinds of trials that we run. So I will share with you what our experience has been with NCI trials. Okay, and of course, I'll give a disclaimer first that we had to make a lot of decisions very quickly. And I'm not saying that anything that I'm discussing today that we had to implement to address the COVID situation will necessarily be adopted on a permanent basis. And so just appreciate that as I go through some of these issues. I'm going to hit on 4 major topics. One is the immediate consequences of the pandemic on our trials, modifications to trial procedures we had to make, discuss in sort of general terms what is the impact on study design and feasibility of some of our trials. I won't get into the level of detail of discussing specific changes to the design or analysis that we might eventually have to make. Also, I thought it was worth pointing out that because of the way NCI has its trials set up, we have standing infrastructure for running our cancer clinical trials. And we actually were able to leverage that to contribute to increasing knowledge about COVID-19 biology and its clinical impact, particularly, of course, on our patients who have cancer. So some immediate consequences is we saw the trial accrual take a nosedive. I'll show you a plot on the next slide. And they differed somewhat depending on the step in the trial, which I'll explain in a minute. Many of our clinics were closed or reduced hours and staffing for in-person visits. Patients were unable or unwilling to come to the clinical trial sites for their clinical evaluations, treatment delivery, specimen collections, etc. And this is, I think, particularly a problem in cancer therapy because many of our patients, due to their treatment, are immune compromised. But also, many of our trials are for patients with advanced disease who may have basically exhausted other options of therapy. So if we shut down the trials, then they may not have any therapy options left open to them. Many sites were pausing the opening of new studies. And again, this is a problem for patients who might have exhausted their options and are looking for something in a clinical trial as sometimes a last hope. So this is what happened to our accrual. You can see that right around mid-March, it took a nosedive. Our accrual reduced by around 50%. We're seeing some bit of a rebound now, but the updated data in the last 2 weeks or so, we don't have yet, or at least I didn't have access to it to present it. I suspect accrual will be going down again somewhat, but we will see. And the 2 different lines that you see there, a feature of many clinical trials nowadays in cancer is that we have genomically guided therapies so that patients are first screened to have their tumors assessed for different genetic mutations, and that will determine whether they are eligible for a trial or not, because many of our trials now have what we call targeted therapies. So you can see that while there was some impact on the screening steps, the the major impact was really on the steps involving the actual intervention with the treatment. So some things that we had to do immediately, I would say that some of my colleagues got very little sleep during the month of March, trying to address all of the urgent questions we had coming in from our clinical trial sites. So we had to make decisions very quickly about what kinds of requirements we could relax. And here are a couple of them. By the second week in March, we were already allowing patients to have their care transferred to a different participating study site. We have, many NCI-funded cancer centers, so it's not uncommon for patients with cancer to travel quite a distance to a major cancer center to get their care, as opposed to being treated locally. Sometimes it would be a clinical site that was closer to their home that would make it easier for them to travel, perhaps not having to fly, for example. So we implemented those kinds of flexibilities when possible. Also, since people were rapidly switching to telehealth, or again, didn't want to have to travel, we instituted some flexibility so that patients could have some of their routine evaluations done by, say, their primary care provider when that was considered acceptable. And things like routine labs, physical exams, vitals, some things that wouldn't necessarily have to be done by an official clinical trial investigator. However, it was important that there was good communication between the study PIs and these local providers to make sure that they understood the assessments they needed to make and could have any questions answered that they might have. Another important change was that normally any of our agents that are being used in trials under INDs have to be picked up by the patient at the clinical site, or maybe the pharmacy at the clinical site. We actually got permission to mail oral agents directly to patients, avoiding their having to come into a clinic, risk exposure to COVID, in order to just pick up their pills, basically. About 10 days later, we had some additional elaborations on these guidance. One thing that we found that was causing a lot of angst is many of these minor flexibilities that we were giving now would technically be considered protocol violations. So we had to give our sites some guidance on how they should track these protocol deviations, and always reassuring them that, patient safety, safety of the clinical staff, patients' families were really foremost important, and that people had to do what they had to do to be safe. So thinking ahead about what some of these flexibilities were going to imply downstream is we know that in cancer, many of our patient groups are relatively small cohorts. So we have a lot of rare cancers, and we only, in our adult trials, we're only successful in accruing into trials, a few percentage of some of those adult cancers. So some of these trials already were kind of at their limit in terms of what was going to be feasible to accrue in a reasonable period of time. This is something we're going to have to come to grips with, whether we're able to extend the accrual period of the trial to counterbalance the drops of accrual, or it's possible there could be some trials that simply become infeasible because of the hit we took to accrual. These will be decided, of course, on a case-by-case basis. Timing and reliability of disease status measurements, adverse event assessments, etc. We have to recognize might have the timing disrupted. It may be that a different laboratory test would be used because that's the only one that was available locally versus the clinical trial site. So all of these issues are going to have to be examined when we do analysis of these studies, considering things as Nancy and Dean have already described, things like special sensitivity analyses, possibly eliminating data from certain sites if it turns out that they simply couldn't adhere to study requirements because of their special circumstances dealing with COVID. Another very interesting thing we encountered that generated a lot of discussion was a change in the risk-benefit balance related to trial participation, and I'm going to elaborate on that shortly. So I already went into some of this. The accrual targets might no longer be feasible for some of these trials. We could try to open new clinical trial sites. We'll have to, again, consider that in the analysis, because then there may be some time differences for which sites were accruing at what times. If there's any drift in the patient cohort because of that, that might have to be considered in the analyses. Another possible change that could be made in some trials is a change in primary endpoint. For example, many of our advanced disease trials use progression-free survival, which requires periodic imaging and assessments that can be somewhat subjective. So there could be potential changes in primary endpoint to sort of harder endpoints like overall survival. We should also consider, and I think Nancy already mentioned this, whether the reduction in accrual is something we'll just have to live with, and will the reduction in statistical power or precision be something that we think still would be sufficient to give us some meaningful results. The timing and reliability of disease status assessments, adverse events, etc. As I mentioned, there could be a lot more wiggle in those things. The best we can do there is to make sure that it's understood that study investigators are still responsible for communicating with local care providers, being available to answer their questions. We can provide additional training materials, things like specimen collection kits. You know, we do a lot of collection of blood samples and tumor specimens, particularly with tumor specimens, if you're going to be using them for genomic testing. There are good ways and bad ways to collect those tumor specimens. And so all of these instructions and actual kits for doing the collection needed to be disseminated more broadly if additional sites were going to be coming on board, or people were going to attempt to do this with their local care providers. Also, we can consider what laboratory assessments are robust. You know, in cardiology, for example, we know things like cholesterol are pretty well standardized, sodium measurements, that kind of thing. In cancer, we have a lot more biomarkers that I would consider in sort of the investigational stage, and it may matter a lot where those specimens are, actually how they're acquired even, and how they're analyzed, what assay protocols are used. So that'll all have to be considered at analysis time. And I think, again, the analysis is going to have to do things like sensitivity analyses and figure out what really has an impact and what doesn't. So this story of this one particular trial, and I'm intentionally not giving you a lot of detail about it because it's still heavily under discussion. There are very strong feelings on all sides of this. But we had a trial in patients with advanced cancer. It was a stratified, randomized, placebo-controlled trial assessing the value of a new agent. And in particular, what was especially interesting in this trial was the value of what we call maintenance therapy. So normally, cancer therapy is given in a number of cycles, usually 3-week or 4-week cycles that might go on for 4 or 5 months. And then the patient is finished with treatment until they show signs of progression, or if we're lucky, they do fine and never need more treatment. So placebo, a maintenance treatment is something that has been gaining a lot of interest lately. So this particular trial was evaluating whether adding this new agent, both as part of the chemo cocktail, as well as including it in the maintenance phase for 20 months, would lead to benefits for this patient population. And it's important to note 2 things: that this particular therapy is delivered by IV infusion. So it really has to be delivered in a special clinical site that has the ability to deliver chemotherapy or cancer-directed therapies such as this one. Also keeping in mind that the standard chemos themselves may suppress the immune system, and the particular investigational agent under consideration here actually also has associated with it immune-related toxicities. So in effect, patients who were being randomized to the placebo arm in this trial were being asked to come in for an additional 20 months to obtain an IV infusion, which if they were in the placebo arm was in fact a placebo. So there was a lot of debate immediately about whether this was appropriate in the era of COVID, when you were asking patients to travel to the clinic for as much as 20 months. We don't know how long this pandemic is really going to be a huge issue, but they would be coming in for as long as potentially 20 months in order to get this placebo infusion. So there were strong feelings on all sides. Many felt we didn't want to disrupt the trial, and that patients, always had the option to drop out of the trial and be unblinded. Others felt that the trial should be changed so that it was unblinded, but we could still continue the trial, but switch instead of a progression-free survival endpoint, which, as I mentioned, is subject to some wiggle in interpretation, that we would switch to overall survival, which is a hard endpoint. And I will tell you that this has not been resolved. There are still ongoing conference calls and discussions involving the IRB, the study investigators. We had a pharma company involved here. The NCI has some pretty strong opinions about this as well. So meanwhile, the accrual has been suspended until we can sort this out and figure out whether we're going to unblind everybody. But this is just one example of how an unanticipated change in the landscape can totally upset a risk-benefit balance that you thought you had well formulated. So some of the discussions, some of the options, as I mentioned, are that we could continue as is and let each patient decide. There was some concern that this might be seen as potentially coercive, saying, well, if you're not willing to keep coming in, then you have to drop out of the trial. We could completely unblind the trial, which would probably require a change to primary endpoint of overall survival. This had even broader implications in that if we changed to overall survival, we would have fewer events than progression-free survival events, and this may lead to an underpowered study or simply make the study infeasible to complete in a reasonable time period. There was also an option floated that we could allow choice of unblinding by clinical site. I think that this will still be a very challenging kind of analysis, but it is an option that as far as I know, has not been eliminated yet. So moving on to how this has changed what our investigators do, we rapidly put out some announcements of funding opportunities for cancer research who wanted to contribute to COVID-19 research. There's sort of a special relationship between NCI and NIAID. We both have interest in viral-related diseases. We have a number of virus-caused cancers now. Immunotherapy is a huge area of interest in oncology right now, and of course that has overlap with NIAID's interest as well. We have shared resources for developing vaccines, both cancer vaccines as well as vaccines for infectious diseases. So it was really a natural kind of marriage. And so some of our cancer researchers can move quite easily over into some immunology type research. And we wanted to make that possible, especially when some of these researchers might not have been able to continue on with some of their cancer trials or other cancer research because of shutdowns due to COVID. So we wanted to try and make lemonade out of lemons by doing the best we could to really bring to bear all the expertise that we had in the cancer community to fight the COVID epidemic. We were also able to leverage our NCI clinical trials infrastructure. We launched a big longitudinal study that is being run through our existing NCI trials networks. Since it was already set up as standing structure, we had the ability to quickly develop databases with online data entry, ability to collect specimens. We have biospecimen repositories. So really we were set up to move quickly and help with the COVID effort. And so that study, the longitudinal study I have mentioned there, we went from study idea to study launch in 6 weeks, which is absolutely remarkable considering our average studies usually take at least a year, sometimes 2 years or more to get launched. So we went from nothing to a launch study in 6 weeks. We also have a trial that is repurposing a drug used in oncology to combat serious COVID-19 symptoms. With immunotherapies, we have a phenomenon known as cytokine storm, where basically the immune system goes crazy. And we have some drugs, one of them being tocilizumab, which has now been put into some clinical trials to fight the severe adverse immune reactions that some patients are having from COVID infection. So just to summarize, I think there are a lot of lessons to be learned about the necessity of some procedures that we think of standard approaches, and we're seeing this even in routine clinical care. You know, one of the biggest examples, of course, is the use of telehealth. You know, could we really reach more patients and do things more efficiently if we use telehealth more? What are the downsides of that? What are we potentially missing by using telehealth assessments? As I mentioned, this has raised the idea of some novel aspects to consider in benefit risk assessment for clinical trials. I think it's emphasized that we need to be attentive and nimble to adapt to continuously evolving landscapes. I know NIAID has examples of studies where they've had fires at clinical sites and they've had to adapt very quickly to salvage what they could out of the trial while protecting patients. So we could probably all be better about that. Sometimes our clinical trial structures are thought to be a little clunky and slow moving, and we need to be able to be nimble. I think it was very instructive to see how quickly we could launch these COVID studies. You have to wonder if we can do this in 6 weeks. Why does it take us a year, 2 years, 3 years sometimes to get our studies launched? And I think we can learn from that. And we're going into this with eyes wide open. We know that there are going to be challenges in the analysis and interpretation of some of our trials, and there's going to be a lot more work to be done as we grapple with these issues. And I would just like to thank my colleagues who helped provide some background information for this presentation. Thank you very much.
Thank you, Lisa. We have some questions and answers in the QA box, and you're encouraged to submit some more in there if you have questions and answers. Jim's going to moderate the question and answer portion of this panel. So I will mute myself.
Okay, well, why don't we begin with one question related to, I think Nancy Geller said, clinical trials may never be the same. And could you expand on that? Are you referring to trials that are interrupted now or disrupted? Or are you talking about the permanent changes that might result?
Well, I think it was really meant to be a pretty general statement. I think while we're in the pandemic, accrual is very difficult. And I don't think that clinical trials are, entering clinical trials are in favor in the general population. And I don't think this helps. So, I mean, we have really, really important questions to answer, and we really need a publicist to extol the benefits of entering clinical trials, participating in them. I mean, for now, I'm certainly optimistic about the vaccine trials, but I think people who enter the vaccine trials don't realize that once they enter a vaccine trial, they can't enter another one. But anyway, be that as it may, I really worry about accrual to clinical trials. Clinical trials may never be the same also means, I think, reliance more on telemedicine than we have in the past. I think that's really a good change, and it will lessen the cost of clinical trials. I think it's really meant to be a pretty broad statement.
Thank you. Well, I think Lisa ended her presentation with discussion of some of the positive, I call them silver linings of the COVID, both what we can learn when we have to move fast, when we have to be nimble, and when obstacles arise. So I think it would be interesting to hear maybe from all the speakers whether there are other benefits that have been learned that you could expand on. I think Dean had to step out of the panel for a moment because of trial-related issues.
Would you like me to comment on that, Jim?
Sure.
Okay, so I agree with Nancy's comments and what Jim just said, that I think there's a lot to learn from what's really important in terms of trial procedures and maybe what is not so important and has been bogging us down. You know, in NCI, we have sort of two opposing forces. One is our trials in some ways are becoming more complex because we're doing all of this genomic testing to tailor therapy to the genomics of a patient's tumor. But on the other hand, you know, we easily get bogged down in all kinds of reporting requirements for, you know, doing all kinds of laboratory assessments and other kinds of clinical assessments and having patients travel to cancer centers. And we reinvented our trials system about, I guess it's been maybe close to 10 years ago now, where we expanded the network and made it easier for even some local community oncology sites to participate in our trials. And I think that this pandemic has really emphasized that that is an important direction to keep moving. And this idea of having more sort of large, simple trials instead of all these data-intensive trials where, you know, unfortunately for cancer patients, especially patients with advanced cancer, you know, survival means a lot. I mean, do we care so much about the little details of tumor size measurements and all of this? I mean, ultimately the patient wants to survive and the patient wants to have reasonable quality of life surviving. So maybe we are going a little bit overboard with some of these many data collections that we're doing in clinical trials. It just adds a whole bunch of baggage, and it is not always clear what the real return from that is. And it would also help with launching the trials more quickly. And if you don't have a zillion data forms to work out and all of that, I think it just keeping things simple, there's a lot of advantage to that.
Thank you. Here's a question from William Rosenberger for, I think for Nancy Geller. What's the impact of a COVID diagnosis in a trial volunteer on heart, lung, and blood? Should those patients be excluded?
Well, certainly not post hoc. I agree with Dean that randomization is holy and should not be violated. I haven't thought that much about the heart trials. I mean, you have the same sensitivity analyses as I mentioned for BMT-CTN, but when you have cause-specific death as an endpoint, you have this problem of whether the death is from COVID or from heart disease is perhaps in the eye of the beholder. And it's something you can do a sensitivity analysis about. Or you could change your endpoint, as Lisa suggested, to all-cause mortality. It's a hard one, and I think it might be trial-specific.
Okay, here's a related question. If recruitment is put on hold, the second stage of a trial may be younger or with fewer comorbidities, since at-risk patients may be more unlikely to enroll. How do you analyze second stages of a trial with different subject characteristics?
Yeah, and I did mention that in my talk. I think that with some of these trials, we will have to look at different time periods and how cohorts might have differed over time and whether that had an impact on treatment comparisons. And this is not uncommon in oncology trials anyway. We do find that in many of our trials, many of our early phase trials actually are single arm trials, which I know many of the rest of the disease areas are not fond of. But But we do routinely see drift in the prognostic characteristics of patients, even aside from any pandemic. So this is something that randomization, of course, can address. But now when you have these characteristics that might actually impact on the treatment effect magnitude, then you have to worry about it a lot, even in a randomized trial.
Okay.
Jim, we missed the first question, which was from Chris Jennison, and it was to me about the planned trial, and it's missed because I answered that I'd answer it.
Go for
In
it.
the chat. I think the question was, in the planned trial, would you, if the results are positive, would you recommend face-to-face or online treatment? Well, the first thing to do is look for consistency. And of course, a test for interaction is not going to be powerful. So assuming you have consistency of direction of results, I think you'd recommend online treatment in this situation because it's so much easier for the subjects, and also it's much cheaper. So I think, and it would be a major result that this technique, which was developed by one of the investigators, could be used in a virtual setting. And I guess while I'm on the subject, there's another question about whether everybody had computers. Turns out most people have computers, and at the worst, and almost everybody has a cell phone, and so FaceTime was in use as well. I didn't write that down, but there may be some that are done only by telephone. I'm not sure, and I have no idea how effective that would be.
Okay, thank you. Here's another question that's a bit broader. Do you think there's scope here for an increase in the use of modeling, artificial intelligence, and other methods to reduce the need for in-person participation? I know there's limits to these approaches, of course, but for drug repurposing, et cetera, it might be a more viable approach than large clinical trials. That's directed at all panelists. And unfortunately, Dean is still not back.
Right. I'll speak for Dean. We think randomization is holy. There's nothing like a randomized clinical trial. I don't, I think artificial intelligence approaches will be very interesting for some of these sensitivity analyses, for maybe secondary analyses. I have nothing against it, but it doesn't take the place of a large randomized clinical trial or of a randomized clinical trial in general.
Yeah, I would, this is Lisa, I would agree with Nancy. I don't think it's going to replace our randomized clinical trials. It's possible that devices could be developed that will help with data collection. So for example, if you have a patient you're interviewing for quality of life variables, could that be done online? Does that require a visit to the clinic to fill out a checklist? There's a lot of talk about wearable monitors. I'm sure Nancy hears about that a lot in Heart, Lung, and Blood. So I think there are definitely ways that innovation could be used to speed up or make easier how we do some of our trials. But I would again emphasize that I'm not a big proponent of saying we can get rid of randomization. I think those kinds of analyses done on existing data sets can be useful as sensitivity analyses. They can be useful to bring in supplementary information about special subgroups of patients for whom you might not have been able to accrue sufficient numbers in a randomized trial. But I'm not ready to give up on randomized clinical trials.
Okay, another question. Here's one from Aldana Rosso, who will be speaking next week. She's addressing this to Nancy and Dean. Unfortunately, he's still not here. You are planning for several sensitivity analyses based on censoring, excluding the COVID periods. This sounds like you want to estimate the treatment effect in a world where COVID-19 does not exist. Is that correct?
Well, I told you my talk has more questions than answers, and I'm not sure I know the answer to this. I think if you maintain the primary analysis as planned, you're not dealing with a world in which COVID-19 doesn't exist. And certainly a world in which COVID-19 doesn't exist is of interest, in fact, would be preferred. So that's certainly something you do want to answer. I mean, I think, you know, what's happening is we talk about more and more sensitivity analyses and more questions about what is the right estimand for a particular trial. We get into issues of multiplicity like we've never had before. But I think each trial will have to answer whether its primary analysis is for a world in which COVID-19 does not exist. And I think, as I said earlier, maintaining the primary analysis as planned does not assume that. It assumes it existed for a while, but then it stopped. So, or that the pandemic started. That's not a very good answer, I acknowledge.
Oh, well, these are challenges that need to be addressed. Let me maybe have time for one more question. And this was directed at Lisa from Mary Lucas. And the question is, you mentioned that you were able to get a clinical trial from the idea to execution in 6 weeks. Is this something that will be reproducible in future trials to accelerate research?
Well, I would say, given how little sleep the trial team got in the month of March, probably not something we necessarily want to reproduce exactly in the future. But I think it does speak to efficiencies of having a big standing trial structure. We have actually a central data system where even our clinical trials that are run through our network, they go into a common system and enter data. The data forms are constructed centrally in collaboration with the group that is actually running the trial. So we were able to leverage a lot of existing things. We were able to leverage this whole data entry system, database. We were able to leverage data forms that had similar variables that were collected on this COVID study versus some of our other trials. There was a huge involvement from NCI staff. I think in terms of having standing statistical groups in our institutes, I think this may sound self-serving, but I think it's very, very important that we statisticians and the clinicians in-house at NCI who are normally doing more oversight and statistical methodology work, all were brought to bear on this issue to get this trial up and running in 6 weeks. We had daily phone calls, people working late into the night, every single night on weekends, and everybody just put 100% effort into this. So while I wouldn't want to necessarily say we could do that on a regular basis, I think we did learn some things about the importance of having these standing structures, these standing groups with the expertise, and be able to reuse things that we'd already developed. And I think that sometimes there's a lot of fussing about details in our trials that maybe is not that productive. And I can tell you numerous times on our calls, I said to people, look, we have to keep it simple. We can't collect every data variable under the sun. You're not going to get people to even bother doing this at all if you give them too much work to do, and you're not going to get reliable data if you insist on having 500 data variables collected. So people had to make compromises. And again, it goes back to what I was saying about sort of the large, simple trials. Figure out what is really important to collect and get it done.
Thank you. I see the time is up, and I don't know if Nancy, you have any final word, but I want to thank all three speakers. I meant Nancy Flournoy.
Yes.
Thank all three of you for participating. I want to mention that a week from today, we have day 2 of this forum and 3 more distinguished speakers. So again, thank you. And Nancy, the final word for you.
Well, thank everyone for coming, and remember you're already signed up to participate in the next forum, and we will be following up with additional information about the more technical sessions that will occur after JSM. So look forward to seeing you next week, and stay tuned.
Thank
Thanks to
you.
all the speakers, for sure.
Bye.
Stay well, everyone.
Yes, you too.